Let's start with the now it's working. Good, good. I am start recording right now. Let's take a pen and paper and let's start with the lab practical. What I want you to study. Lab practical. Lab practicals will have lab 8 to lab 13. Lab 8 to lab 13. Actually, lab 8 is not because we work on it a lot. So it's let me go to the lab. I think I am not covering lab 8 in the exam. Let me start it. You have to understand very well the meiosis, very, very well. All the steps, how many cells we obtain at the end, how are they? Are they haploid, diploid? You have to understand the cell cycle. The cells spend most of their time at 90% of her time, they're in the interface. 10% is in the mitosis. Centrioles, the one that are going to be the source to make the spindle, mitotic spindle, they are only in animal cells. They cannot be found in the plant cells. The cleavage pharaoh happen only, it's the way that the cytokinosis or separation of the cytoplasm happen in animal cells. What are chromosomes homologous? When the chromosome homologous start pairing with each other and when they start separated from each other, chromosome homologous, you have to know all the pictures of the mitosis, animal as well as the plant cells. Prophase, how it look like for both animals and plant, metaphase, anaphase, telophase, and cytokinosis. Remember that for eukaryotic somatic cells, because any cells that is inside of our body, it's somatic except our sexual cells, testes and ovaries. All those somatic cells except testes and ovaries, they will go through what we call mitosis. Mitosis is giving me an equal, the same nuclear division. I start with one cells with 4 chromosomes. For example, I end up having 2 daughter cells that they are identical to each other with 4 chromosomes and they are identical to the original cells. So it's an equal nuclear divisions. I did a post, a chromatography, a pigment. So I want you to know the names of those pigments. Carotene, xanthophyll, chlorophyll A, chlorophyll B. The spectrum, you have to learn the spectrum go from 354 10 nanometers, which is a blue violet color, to red, which is 750 nanometers. You have to understand the meiosis. Meiosis 1, meiosis 2. Meiosis 1, meiosis 2. Remember that meiosis is also preceded by an interphase. An interphase means that they have also an S phase there where the chromosome get replicated to chromatid sister. So they're in meiosis when the chromatid sister got separated. Understand the structure of a chromosome with their centromere. How do chromatid sisters look like? All the phases, what happened during the prophase, metaphase, anaphase, telophase, what happened? Meiosis, prophase 1, what happened? Prophase 2, metaphase, prophase 2, exactly, they're in meiosis 2. Metaphase 1, they're in meiosis 1. What's happened? Metaphase 2, they're in meiosis 2. What's happened? Anaphase 1, what's happened? Anaphase 2, what's happened? Telophase 1 and cytokinosis, telophase 2 and cytokinosis. The structures, anatomy structure for a chloroplast. What we call the organism that capture the sunlight and synthesize carbohydrate from CO2. We call them autotroph. Spermatogenesis. What is it? What is giving me? Oogenesis. What is it? What's giving me? What are the raw material? What the global equation for photosynthesis, the global equation for cellular respirations? Once a sperm is fertilizing an egg, what we call this egg, this single cells that has been fertilized by the sperms, what we call it? There will be a lot of pictures of several all the phases of mitosis, meiosis. Get familiar with it. The leaf that we study with the one that store the starch, we use iodine and it appear dark purple. Which light? A chlorophyll, those pigments that they are in the chloroplast, they are absorbed. Which light? The blue and the red. The blue or violet. The green, yellow, and orange are not absorbed. They are not. I show you pictures of those experiments that we are boiling with water, the leaf, and we boil it also with 95% ethanol. Why we do that? What is the purpose for doing that? It's to remove the pigment. All the phases for the cell cycle. When we do a chromatography, the paper that do this chromatography, it's display a pigment, different pigment. We call those pigments chlorophyll A, chlorophyll B, xanthophyll, carotene. We call them chromatogram. Chromatogram. Crossing over when it's happened. And what we call the process that caused the crossing over, the synapses. And it happened when? It happened during the prophase 1. The piece of equipment that we use to separate, and this you see it in the virtual labs, and we talk about it also there in the lab. The piece of equipment use a prism PRSM to separate white light into its compound of wavelength. What we call it? We call it spectroscope. This spectroscope. spectroscope. That's the piece of equipment that we use to separate the white light into compound wavelength. The chromatography, those two pigments that we have carotene, we have chlorophyll, we have xanthophyll, we have chlorophyll A, chlorophyll B. This, we called out all those pigments, we call them, I didn't have any more space. I have to raise. We call that chromatogram. Those pigments are called chromatogram. So the paper that display those pigments is called chromatogram. This is the chromatography paper, the one that display all those pigments. This is the machine or the piece of instrument that we use to separate all the light from the white light. This is the paper, chromatography paper, that we use to separate all the pigments. You see the differences? That's all for the lab. Any questions? You can start, either send me an email or right now or text. Any question? Okay, great. So we are going to move to the exam 3 because we finished the unit 3. What I want you to study for exam 3. That's for the lab. When I hear the click, that means somebody is texting me. Hold on a minute. I will be there. Exam 3. Cancel. How much time for the practical? 50 minutes for 50 questions, or maybe less. I didn't put the time yet. Exam 3. Cell membrane, structure, function. All the compound of the cell membrane, their job, the phospholipid, the protein, the cholesterol, their function. Net diffusions, what is it? What is the difference between osmosis and dialysis? What is the difference between osmosis and dialysis? Very simple. Osmosis is movement of water. Dialysis is the movement of the solid itself. It's not only the water that moves, but the solid that moves. That's dialysis. You have to understand what is hypertonic, hypotonic, and isotonic. Difference in osmosis in contact of cell animal in the animal cells as well as in the plant cells. Difference between passive transport and active transport. You have to understand the most important one that we study, which is the sodium pump active transport, how it's work. The other active transport like pinocytosis, all the endocytosis and exocytosis, the three of them, phagocytosis, pinocytosis, receptor-mediated endocytosis, cellular respirations. Know it very well. Now the general equations. What is this metabolism? Is it anabolic, catabolic, and endergonic, exergonic, delta G? Is it negative, positive? How it's happened, where it's happened. There are two types, aerobic, presence of oxygen, anaerobic. What's the common between aerobic and anaerobic? They have one thing in common. They have. When equations really happen in both, either they are aerobic or anaerobic, when metabolism happen, which is the glycolysis, the splitting of glucose, happen with or without the oxygen. What's mean redox process? How during chemiosmosis, what's happened? What's happened exactly and where it's happened? Where the electron transporter proteins exist? They're in the cellular respiration. Where are they? Who is the donor of electron and who is the final acceptor of electrons? The donor, they're in the cellular respiration, is the organic fuel, which is just a molecule of glycose we make in it, and the final acceptor is your oxygen to make a water. The two process how the ATP is formed during the cellular respiration. One is by substrate level of phosphorylation, where it's happened and how. And then the oxidative phosphorylation, where it's happened and how. Difference between reductions reactions, oxidations reactions. Know all the steps for cellular respirations. ATP is always produced by phosphorylating ADP, but there are two types of phosphorylations. Substrate level oxidative, you have to understand how. I already talked about this, but yeah, I insist on that. Why in the earth we are doing all the steps from the food arriving to the oxygen? Why? We go from an electron that is highly energetic to lower energy. So we are like going from high state of energy to low energy for the electrons. So the oxygen will accept the low energy electrons. Cellular respirations, we making it very simple by using as organic fuel glucose, but it could be fat, it could be proteins, it could be polysaccharides. All right. We have a case that we call the amination of amino acid. The aminations in amino acid in mammals yield an amino group that we call urea. And this urea is extracted and carbon chain are converted to one of the reactant of glycolysis or the citric acid cycles. The two type of fermentation that we study, alcohol and lactic anaerobic fermentations. We saw that in the lab more, but I want you also to look at the spectrums or the wavelengths, starting from the shortest to the highest on wavelengths. So you know that the TV and radio wavelengths are very high as compared as the gamma rays and X-rays and UV and visible light and infrared and microwave. You have to understand that when the wavelength is shorter, the energy is higher. So the TV and radio waves emit higher energy. Their wavelengths, emit lower energy. Their wavelength is very high. The microwave, the same thing. So if I go from shorter wavelengths to higher wavelengths, I will begin with the gamma rays, gamma X-rays, X-rays, gamma rays, gamma, X, UV, visible, infrared, microwave, TV, and radio waves. So when an electron accept a photon coming from the light, he go from a state from low energy, he got excited, an electron. So he got from lower energy to higher energy. The general equation for cellular respiration and chloroplasts. During the photosynthesis, we know that during the photosynthesis, the red and blue light are absorbed. They are absorbed by what? Who absorb them? Those pigments. Those pigments, we call them chlorophylls. They absorb most those wavelengths than the others. How the ATP is formed during photosynthesis? Heat is formed during the light reactions. How? Where those ETC electron transporter chain that are localized during the photosynthesis, where are they? The photosystem 1 and photosystem 2, you have to understand their functions. The first step for the Calvin cycles is start with attachment or fixations of the CO2. What is the molecule responsible and what is the enzyme responsible for it? How it's work? Any questions? That's conclude our exam 3 for unit 3 lecture. Any questions, dear? And Charles, you have, sorry, I forget that you are non-major. You have 60 minutes for each, I think. You have 60 minutes. Hold on. Let me check this, then I go back and see your questions. Hold on. Let me make sure how many minutes you have. Test options. You have 60 minutes for the exam. Let me go back to the lab. Oh, God. Lab practical 2, not 3. 2, you have 60 minutes. That will help you a lot to study. I record this for the simple reason that some of my students were there, but they didn't bring their laptop, and it was my fault. I mean, I decided last minute to go virtual. It's my fault. I supposed to do it, but it's better to do it virtual later review, and we will keep doing this habit, going virtually for reviews. Okay, I'm going to stop the recording right now, and it will be posted as soon